Well, I hope you're having a great weekend, and I thought maybe this would be helpful to record a little bit of information again about enzymes. So this is the finale really of this chapter, chapter 3, where we have, first of all, a definition of what enzymes are and what they do, and then we look at some specifics on how enzymes are regulated within the body. So let's take it away. So enzymes are catalysts. So you can see this word bolded here. And we know that catalysts are things which can help a reaction to happen, but don't enter into the reaction. In other words, they aren't part of the reactants and they aren't the products. And most of those catalysts are proteins. And we talk about enzymes. And then there's one other type, which is called a ribozyme, which is a different type. But for now, we're just going to focus on enzymes. So let's take a look at a reaction, and we're going to look at an exergonic reaction where we see that there's an energy barrier between the reactants and the products. And so let's take a look at the graph because that's the easiest way to talk about it. So on this y-axis, we see the measure of free energy. And so we could look at this in terms of, this would be the Gibbs free energy equation if we wanted to look at this measurement in a different way. But we actually see that over here in that we can see that when we start with these reactants at this level of free energy and we push it toward the product side, we're changing that Gibbs free energy value toward something that has less energy. The products will have less energy. But we'll have more of them, so that we'll increase the entropy of the of that material. So we'll have more entropy if we wanted to look at the Gibbs equation again. But we really aren't. What we're looking at here is this idea that here are the reactants at this level, and we're going to create these products down here. But what we have is a little barrier, and that barrier would be this activation energy barrier, E sub A, which is just energy of activation or activation energy. But we have to, or the body will need to get over this barrier. We're looking at our metabolism in order to create these products. So this is the challenge right here. Now, the reactants can eventually get to this point where they get into this transition state, sort of randomly, and they can form these products without an enzyme. But the enzyme is going to help that happen more quickly. And so we're going to see the activation energy is necessary to get a reaction to move forward, and the enzymes lower that. So they're going to allow reactants to come together. They're going to allow them to react more readily. And you kind of know already the mechanism of that because you understand that enzymes do a number of things. They have an active site, and that active site is going to facilitate the substrate's binding. And anything else that's necessary to move that reaction forward can also interact with that. So here's that example of sucrose. And here's the picture that you want to recognize and understand. So here they're making this look, like here we have a really large activation energy barrier. So this would be an example of perhaps one type of reaction with no enzyme. So that's the uncatalyzed reaction. But when we add the enzyme as a part of the reaction, we see that it lowers that activation energy barrier, facilitating that reaction going forward. Now, one other thing, I'll come back to that. So either way, we're going to get to this product or the products. But the speed at which that happens, the rate at which that happens is much, much faster with an enzyme. Enzymes are also very highly specific. That means that by design, remember they're proteins, they are designed to catalyze a particular chemical reaction. When we look at the things that we call reactants in biology, we look at very often this term substrate. So we're going to talk about substrates, and you just think, okay, those are the reactants. So they're going to bind to the active site, and that enzyme has high specificity. Just means it's really picky about what it's going to allow to go into that active site. Here's a great picture that shows you little graphic of here's sucrase, the enzyme, and it has this active site here. So the active sites don't always look like I draw them, like little Pac-Man mouths or whatever. Here you can see, but that fits this substrate, sucrose. Sucrose enters that active site. And notice what else is happening here. So we have water, the addition of water, so we're going to have hydrolysis here that's going to break the bonds. And now we have glucose and fructose. So when we look at the entropy part of it, remember that's the measure of disorder, we can say that now these two things here, there's higher entropy than there was here, where sucrose was more of an organized molecule. But in the process of doing this, we've released some desirable products, including energy. Here's the enzyme substrate complex model. So you can see enzyme plus the substrate is going to create this complex, meaning that they join together. But then the enzyme goes off and finds another substrate, and the product has been formed. So you don't see any change in the enzyme. It just goes and then finds another substrate. Here's the way it happens. Enzymes will allow the substrate to enter the active site. They may actually, and very often within this active site where I'm showing here, are the amino acids, and their R groups are ones that will interact with any of the composition of the substrate too. So that's part of how this works. So they enter. There's some binding. There could be some rearrangement of the substrate, putting it into a good position. And then the enzyme changes its own shape enough to create a little strain on these bonds of that substrate, which will help the bonds to break, and that will lead us toward that reaction. So this is lowering that activation energy barrier. So substrate orientation. Also, there can be chemical groups, such as the R groups, that are involved. And this model is called the induced fit model, because the enzyme actually changes shape when it binds to its substrate, creating this strain. Here you see it again. So some enzymes require helper molecules, and those can be a variety of things. They can be ions, which are cofactors. They can be coenzymes, which will be involved with doing something with those chemical groups that the substrate has. And this is very broad definition. We'll see a couple of examples coming up. Or it could be a prosthetic group, which is something that doesn't have any amino acid groups on it, but it's something that is actually bound to the enzymes themselves. Here are some of the examples. I think the one I keep talking about, because I think it's somewhat familiar to you, is this prosthetic group heme, as we find in the enzyme hemoglobin. But then there's some coenzymes, very often vitamins, or some derivative of a vitamin, or this ATP, which is actually a source of energy, but we're going to talk about that, and cofactors. So there they are. An enzyme will, as soon as it becomes in the presence of a substrate, as soon as it contacts the substrate, it's going to start picking up the substrate and reacting. But ultimately, there's a point where the enzymes that are available at that moment in time are all occupied. They're all bound as they are doing their jobs. But then when that happens, we say they're saturated. And what you see here on the graph is that idea of saturation. So initially, we get a very quick rate of reaction as the enzymes are starting to pick up substrates. But as soon as all of those enzymes are occupied, we get this flattening out of this curve, which is saturation. You can see it there. Here's the enzymatic the reaction without the enzyme, and you can see how this is much flatter, much lower. It would take a long time to reach what the reaction, the final products of the reaction, much longer than if it has an enzyme. we can gauge what the maximum rate is used to do this kind of activity by looking at the enzyme's efficiency. In other words, how many molecules are actually converted per unit time. And here they give you the range, which is quite large. Let's look at a pathway, though, because in biology and in metabolism, we're going to see not just individual reactions, but metabolic pathways. And a metabolic pathway is going to, well, actually, the next slide won't show it yet, but I will on the following one. The metabolic pathway is important because what it ultimately does is it tries to achieve for that organism what's called homeostasis. Homeostasis, which is a steady state, a stable condition in the organism. I think I used the analogy in one class that if you get really, if you're in a cold room and you don't have a coat or something, you start to shiver and your body will then warm up a little bit. That's the effect of shivering. That's a way of trying to achieve homeostasis when you're cold. Enzymes are a part of this because they can regulate our metabolism. And they can they can turn off activity or they can turn it on. And we're going to see that here in this, I think, one of these next slides. If not, I'm going to show you on the board. I've got to find this here. They do not do a very good job with this. So I'm going to show you when we get into class, but we're going to see the pathways. I think that'll make more sense. Other ways that enzymes get regulated, though, is there can be inhibitors that will slow an enzyme down. These aren't always toxins. These can be natural inhibitors, and we'll look at this in a pathway. But then there are some things which can be inhibitors that are things we don't want, artificial types of inhibitors, which could maybe be pesticides or some toxins. And sometimes we want to use inhibitors if we can, particularly with some diseases. When an inhibitor inhibits, it goes in and it binds. And if it's an irreversible inhibition, meaning it can't go backwards, it's bound permanently, then what happens is that inhibitor is going to covalently bind to some side chain in the active site, and that it inactivates that enzyme. So that's one way in which irreversible inhibition can happen. Here's an example that they gave of irreversible inhibition. So this is an enzyme, and you might say, uh-oh, I never heard of that. Acetylcholine esterase. So acetylcholine is a chemical that's in our body. It's a neurotransmitter. It's a chemical messenger. That's what acetylcholine is. And acetylcholine esterase is the enzyme involved with that. So this is an irreversible inhibitor, this DIPF. And you need to look at this a little bit in your book to understand what it is, but you can see what it does right here. There are also reversible types of inhibition, which is far more common. One is called competitive. We looked at this. And a non-competitive, which is going to actually bind somewhere other than the active site. So the difference is where it's bound. So competitive is it competes with the substrate for the active site, and a non-competitive binds somewhere other than the active site, changing its shape, which was what we call allosteric. Allosteric. And we'll see that hopefully in a second. So competitive inhibition, it's a good picture. We looked at that video. Non-competitive, you can see the inhibitor comes in other than the active site, and in doing so changes the shape of the enzyme. It doesn't always necessarily close it entirely like they're showing here, but it changes it enough that the substrate doesn't fit into its active site very well. So allosteric, here's the term, is when whatever is bound is changing the shape of the enzyme at some other location other than the active site. And allosteric regulators, though, so let me back up a second. So we're talking about allosteric regulation, and why we're not just talking about inhibition here, we're saying that sometimes that change actually activates an enzyme. We're going to see more on that in class. So stay tuned.